How do drugs communicate with cells and produce physiological responses? Welcome to this high-yield explainer. Today, we're unpacking the absolute masterclass on how cells actually translate chemical messages into biological action. If you've ever wondered how a tiny little molecule can completely alter a patient's heart rate or, instantly relieve pain, well, you are in the exact right place. We're going to break down the specialized macromolecules that make all of this possible. Physiological receptors. Everything from those rapid-fire ion channels down to the much slower nuclear transcribers. And just to make sure you're getting the absolute gold standard of medical education here, all of the highly testable concepts we're covering today are pulled directly from the authoritative text Rang and Dale's Pharmacology, 9th edition. We've distilled the core structural and functional families of these receptors into a highly visual format. It's specifically optimized to help you absolutely crush a 25-mark pharmacology long-answer question. All right, here is our roadmap for today. We've got drug receptor basics, type 1 ligand-gated ion channels, type 2 GPCRs, type 3 CAN-linked receptors, and finally, some exam pearls and revision. Okay, section 1, drug receptor basic concepts. Let's get right into it. So, right off the bat, notice the absolutely crucial separation between the act of binding and the act of actually triggering a response. You have to differentiate these two fundamental terms for your exams. Think of the receptor as a lock, right? Affinity dictates if a drug, which is our key, will physically fit and bind to that lock. But efficacy, that determines if the key can actually turn and open the door, activating the receptor to kickstart a biological change. Now, since efficacy is kind of a sliding scale, it's a graded property, not all agonists are created equal. Full agonists possess high efficacy. When they bind, they are fully capable of producing 100% maximal tissue response. On the flip side, partial agonists only have intermediate efficacy. That means even if they saturate and occupy every single receptor on a cell, they can literally only ever produce a submaximal or partial response. And please pay very close attention to antagonists, because these are massive in clinical pharmacology. Antagonists have fantastic affinity. They bind perfectly to the receptor. But their efficacy, it's mathematically zero, meaning they occupy the receptor and just sit there, effectively blocking it. They prevent any endogenous mediators or full agonists from getting in, acting as this vital functional blockade without causing any activation themselves. Moving right along to section 2, type 1 ligand-gated ion channels. When we talk about these type 1 receptors, we are talking about absolute lightning speed. As the textbook points out, the sheer speed of this response, which literally happens in just a fraction of a millisecond, proves there's no time for intermediate biochemical steps. The coupling between the receptor binding site and the ion channel is totally 100% direct. Picture this structurally. They're usually heteromeric assemblies, made up of 4 or 5 individual protein subunits. These subunits perfectly lock together to form transmembrane helices that surround a central aqueous pore. The exact millisecond a ligand binds, that pore pops open, acting as a rapid gateway for ions like sodium, potassium, or calcium to just rush in or out of the cell. You definitely want to commit these classic examples to memory for your exam, for sure. Why? Because they're responsible for the absolute fastest synaptic events in the human body. The nicotinic acetylcholine receptor handles rapid neuromuscular transmission, while the GABA-A and NMDA glutamate receptors mediate those incredibly fast inhibitory and excitatory signals in the central nervous system. All right, section 3. This is a massive one. Type 2 G-protein coupled receptors, or GPCRs. The absolute crucial point here is recognizing their unique architecture. The defining structural signature of a GPCR is its heptahelical design. Hepta meaning 7, right? So this means the receptor consists of a single continuous polypeptide chain that literally weaves back and forth through the cell membrane exactly 7 times. Now, unlike the direct millisecond-fast ion channels we just looked at, GPCR signaling is a multi-step, moderate-speed cascade. Step 1, the agonist binds. Step 2, this triggers a really profound conformational change inside the cell, causing a GDP to GTP exchange on the G protein. Step 3, this newly active G protein splits into an alpha GTP subunit and a beta gamma complex. And finally, step 4, these active subunits go off hunting to associate with their specific cellular targets, like enzymes or other channels, to drastically amplify the signal. This table right here, absolute prime exam material. You must know that the G subtype stimulates adenylocyclase to increase cyclic AMP.
The GI subtype does the exact opposite. It completely inhibits adenylyl cyclase. And the GQ subtype? Well, that activates an entirely different pathway called phospholipase C, which spikes intracellular calcium. Super familiar therapeutic targets like adrenergic and muscarinic receptors? Yeah, they all operate through these exact GPCR pathways.
Next up, Section 4, Type 3, kinase-linked receptors. These are a completely different beast compared to those 7-pass GPCRs. These receptors typically just feature a single membrane-spanning helix. They've got this massive extracellular domain to catch really large protein ligands, and an intracellular domain that is just packed with enzyme activity. And because they ultimately regulate things like gene transcription and protein synthesis, their effects are way slower. We're talking hours instead of milliseconds, and they profoundly dictate cell division, growth, and metabolism. Some seriously important physiological mediators rely entirely on these type 3 receptors to orchestrate long-term responses in the body. The absolute most famous example here has to be the insulin receptor, which acts via a tyrosine kinase phosphorylation cascade. Growth factors, inflammatory cytokines, metabolic hormones like leptin, they all use this slower, enzyme-linked architecture to achieve their massive cellular changes. All right, let's bring it all together in Section 5, Exam Pearls and Revision. Go ahead and take a mental snapshot of this summary table. It beautifully contrasts the 3 main cell membrane families. Oh, and for your exams, definitely don't forget the 4th family, intracellular or nuclear receptors, like steroid receptors, which aren't on the membrane at all and take days to alter gene transcription. But for these top 3, really notice the evolution here. The complex 4 to 5 subunit structure of the millisecond-fast ligand-gated channels to the 7 transmembrane GPCRs that operate in seconds to minutes, right down to the single helix kinase-linked receptors that dictate metabolic changes over several hours. Okay, let's test your active recall. Imagine you're sitting right across from your professor in Aviva right now. Which of these receptor families is responsible for the absolute fastest synaptic events? If you said type 1 ligand-gated ion channels, boom, you nailed it. But make sure you also tell them the why. It is their direct coupling mechanism. There are no middleman G proteins or enzymes, and that's what allows for a response in a mere fraction of a millisecond. Here's another classic long answer prompt you are almost guaranteed to see. What is the unique, defining structural feature of GPCRs? The magic phrase your examiner is desperately looking for is the heptahelical structure. You must be able to confidently explain that they comprise a single, continuous polypeptide chain that snakes through the cell membrane exactly 7 times. Remember, physiological receptors are classified into ligand-gated ion channels, GPCRs, enzyme-linked receptors, and intracellular receptors, each differing in mechanism, speed, and therapeutic importance. To wrap up our explainer today, I really want to leave you with a final thought to ponder. With the brilliant architecture of these receptor families now completely mapped out in your mind, how does that subtle mathematical property of efficacy that we learned about in Step 1, how does that determine if a drug becomes a life-saving cellular trigger or a vital silent blockade? Truly understanding that intersection of structure and function, well, that is where real pharmacological mastery lies. Keep learning, keep asking questions, and I'll catch you in the next explainer.